Thank you very much for having me here today. So I'll go ahead. I'm a student at Sam Writer's Lab in OIST, and we study the brains and behavior of cephalopods. Yeah, as you said, I'm going to be talking primarily about squid recordings, but I thought first maybe to slightly introduce the motivation why study cephalopods. So cephalopods like octopus, cuttlefish, and squid are invertebrates. Amongst invertebrates, they possess the largest and most complex brains, alongside a range of unique behaviors. Perhaps most noteworthy, if you've never seen before, their ability to camouflage. So they possess these chromatophore cells in their skin, which they can dynamically control with their brain to change color to match the texture or otherwise at once. And these chromatophore system, as well as most of essential systems in a cephalopod's life, are reliant on their vision. So here, if you have a look, this is the cephalopod eye next to the vertebrate eye. They have convergently developed, strikingly similar camera type lens eyes. However, the visual processing system behind these, their vision is completely, seemingly at least, different. The visual system is the part where I focus my research here at OIST. So here you have a look. This is the big fin reef squid, Sepioteuthis lessoniana. This is the model most people in my lab use. And I don't know if you can see okay, now you should be able to see my pointer, I hope.
Yes.
Through their transparent skin in between the eyes, you can actually see the large brain. If we have a closer look, this is a coronal section of the squid brain. You can, I think, see three distinct structures, the central brain in the middle, and then on the left and right, these bean structures are the optic lobes. These are the visual processing centers of the cephalopod brain. And if you pay attention, you can see how it has a very distinct layered structure that if we have a closer look, looks like this. So this superficial layer, often referred to as the deep retina, is where the information from the cephalopod eye, from the retina of the cephalopod eye, directly arrives in the brain, and through here travels through the medulla towards the central brain. So for my project, I've been interested to understand how the function of this circuit, basically, because most of our knowledge thus far has been primarily through anatomy, molecular studies, or electrical stimulation studies. It has been difficult to actually record up until now from the cephalopod brain, with some few exceptions in the past. So to do this, we had to develop, first of all, a new method to head fix a cephalopod like our squid. Needless to say, head fixing a soft-bodied, boneless mollusk has been quite the challenge, but through the efforts of a lot of people in our lab, we have come up with a protocol, and now we're finally using this to actually do recordings using Neuropixels. Here's a top-down view where the probes go through the brain through these, let's say, cranial window. And if you look at the side view, here's the whole experiment is basically happening underwater. So we had to figure out a way to actually waterproof the entire system for the experiment to work. And as I mentioned earlier, cephalopods are in fact soft-bodied. So the stability of the head fixation is not, let's say, it's definitely not as great as you will find in other models. However, if we have a look at the drift traces of these two probes from this example experiment, it is actually decently stable, and we can, in fact, as I'll show you in a second, record from individual neurons for a number of hours. We are still only able to do acute experiments, but up from 3 to 6 hours, after which the quality of the recordings drops substantially. But there's been good and steady progress. So, oh yes, another thing I wanted to point out is actually why the Neuropixels is particularly a great tool for this kind of experiment is because if you have a look here at a scanned sagittal view of the cephalopod optic lobe, actually, these are the traces of these two probes, we can simultaneously record from all the different visual processing layers of the brain and see the neuronal activity in response to visual stimulus simultaneously, which is actually why, for our experiments at least, Neuropixel 1 seems to be even more ideal than even the newer versions. So to actually move ahead and have a look at how the data looks like, here is a small example of the trace. We use in our lab kilosort for the spike sorting. The different colors overlaid on the plot respond to different clusters kilosort has identified, after which we use manual curation with phi. So as you, I think, saw in earlier class yesterday and today, and we identify good channels out of bad or artificial units. And out of these selected units, then we apply a series of automated quality metrics. So we actually use both manual and automated sorting. And you've seen all of this actually today, I'm pretty sure, but basically we look at the waveform shapes and the distribution of the waveform across the channels, the amplitudes, and of course, we check for the autocorrelograms for the presence of any refractory period violations. One thing that I think stands out in my mind is that cephalopod neurons seem to be, the waveform of cephalopod neurons seem to be quite similar to those of vertebrates after all. But I'm not that familiar actually with vertebrate waveforms, so maybe someone later could point out possible differences. So after we sort out all our units, what we want to do is we actually want to map the neurons we record from in the cephalopod brain. So what we do in my lab is we use tissue clearing and light sheet microscopy. We take the scanned brains and annotate the locations, the trajectories the probe took in the brain, as well as the different brain regions. And then using nonlinear warping, we register all of these scanned brains to a common reference atlas, which you can see here. Each line corresponds to a different probe. And now to actually map the units on top of the neurons on top of these trajectories, what we do is we look at the cross-correlation of the LFP signal across all the channels. You can see, I hope here in this plot, how the more superficial channels after 350, there is a very clean band of cross-correlation, high cross-correlation. These are the channels that are in fact outside the brain and are within in seawater. So we can identify the boundary between seawater and brain. And then using this knowledge and the knowledge of the waveform, we can actually, across the channels, we can actually map the units onto the brain atlas. And yeah, so this has been working pretty well. It's still a work in progress in the lab, but it's been going really well, and we can actually use this procedure to actually study the visual system. So I'm going to show you very briefly just two examples if I still have time. One of the experiments we do is a very classical visual stimuli like drifting gratings. Here I have the raster plots and histograms of a single neuron, which you can see is highly selective for gratings traveling downwards and gets suppressed when they travel upwards. If we look at the locations of all the neurons we recorded from in an annotated atlas, then we can see the ratio of direction selective units versus all the units we recorded from. And you can see how there are in fact direction selective units across most layers of the optic lobe, except the very most superficial ones. And then another typical experiment we do is the locally sparse noise, which you can use to identify the receptive field of a neuron in a pipeline such as this. So you can have the receptive field size and location. And then again, with a little more complicated pipeline here, we can look at the atlas annotation, as well as the distance of the neurons we record from the outer granular layer. So the distance of each point in the atlas from the superficial layer of the optic lobe. And if we compute now the size of the receptive fields over the depth on this atlas, you can see that as you go from the very superficial layers deeper in the brain, in the medulla and the peduncle lobe, the size of the receptive field of these neurons increases, indicating a convergence of visual information. So with that in mind, I hope I wasn't too long. I would like to thank my lab, the Computational Neuroethology unit in OIST, and particularly Dr. Mano and Professor Reiter, as well as the technicians at the lab. Thank you very much.